First, I would like to thank Dimitri that invited me to Institute Pasteur Korea. Actually, it's my first time in Korea. We are so close. So I introduce a little bit of myself because as you can be confused. Actually, I'm not French, but I'm working with Institute Pasteur in Paris for more than 20 years. I have a head of this unit in Pasteur, and also we have the CNS mixed unit in enrollment. And also we also a unit of INRAE as well at the same time. So today I would like to talk about to go with the theme of this session is about the effect of ecosystem on the vector-borne infectious disease. But I think you heard a little bit in the morning. But this is I'm a biologist. This is the view from biologists because this morning is from the climatologist. So I think this is a way that you know that see that the world has increased in temperature actually in like 4 degrees Celsius, maybe in the north. So what is interesting about this study, because if you can see between the they compare between the natural driver, which is the temperature in the volcano and the temperature outside, you can see that actually the human or natural other activities that increasing the temperature. So now the question is how we can slow it down, and because of this effect of human. So you heard a lot about this the climate change and health this morning. You can have the direct impact, by flooding, drought, heavy rainfall, and heat wave, or ecosystem-related health impact of change in habitat of insect vectors, mental health, or indirect, with all these losing of means of livelihood, food, and migration, conflict of the effect. But today I'm going to focus on my expertise, which is the insect vector. So the disease that we are working a lot in the past 15 years is the dengue viral infection. So dengue, as you know, is a mosquito-borne viral infection. We have 4 serotypes. Severe dengue is the leading cause of serious hemorrhagic disease. No spec treatment, incidence increasing, but also very interesting. You see that the African seem to be the continent that don't have a lot of dengue outbreak or have problem with dengue. So many reasons about that. And also, we have a mini outbreak in Japan in 2014, and also we have increase of dengue cases in France as well. So the prevention and control of dengue now is depend on vector control. So we have a dengue vaccine, which is not have a limitation of use. But the view of our biologists when we're looking at how climate affect the transmission of dengue is focus on the effect of the temperature on the mosquitoes. So you can see that the way that mosquitoes can bring in transmit the dengue to human, we call vector two things. Vector competence, which is the intrinsic factor of the vector populations. So it depends on whether that mosquito is susceptible to virus. And also, it's not depend only on intrinsic factor of the mosquitoes, but it's also vector capacity, which is including the biting rate, mosquito densities, transmission parameters, and mortality rate, and other extrinsic incubation period, whether they can grow fast or less. And this factor affect by the temperature. So we can do the experiment in the climate chamber, and then to see how the temperature change. So you can see that in figure B, the temperature, when it's increasing the viral vector capacity. But when it is above certain degree, like even above 30 degree, the vector capacity decreasing. So it doesn't mean that when the temperature going up, you have more dengue. You can have less dengue transmission, because that affects the vector competence. Also, it's not only the temperature itself, the absolute one, it's the diurnal variation that is quite important. So like, if the morning is 0 and then the afternoon is 20 degrees, so you have more diurnal variation and you have more transmission. If the baseline temperature is low, like figure D, at the 14 degree. So if you have high diurnal variation, you increase the vector competence. But if the temperature is high, like 26, and then you 26, you're going to have a decrease of vector capacity. So there are so many factors in temperature that affect this transmission. So this is something that if we see that we have increase of a lot of the autochthonous events of double virus in mainland of France. In 2022 only, we have increase of dengue cases up to 60%. So autochthonous means that these dengue cases never go out from France. They get infection in France. So that means the transmission of dengue already occur in French metropolitan, because we know that they have a lot in French Outre-mer. So when we try to do the predictions of dengue epidemics in the world, so we take into account all these parameters of vector capacity. So if we do under the business as usual that we are doing now, we don't change anything. So you can see that the difference between the figure A is about different between 1980, 1990 and the early 1990. So it's increased a lot. But if you compare between early 2000s to the end of this century, so the area that used to have dengue, like Thailand, India, we won't have dengue anymore because the temperature is too high for vector to transmit dengue. But then Korea, Japan, Russia, Europe will be the region for the dengue epidemics. So this is something quite alarming, and then how we can do and prepare for that. So the first thing that we try as Institute Pasteur, we obtain this funding from NIH. So we create the research centers called Pasteur International Center for Research on Emerging Infectious Disease, or PICREAT. So the project that we start is the interregional study of transmission and adaptation and pathogenesis of virus with pandemic potential in Southeast Asia and West and Central Africa. So we acquire. So in this NIH funded CREAT network, so you can see that they have 10 networks, but we are the only one that we don't have any US partner. So we are so proud of that. So we have partner in Pasteur Dhaka, Pasteur Cameroon, and Pasteur Cambodia. So this is the list of our collaborators. You can see that, and then we just add Germany and Turkey because of the CCHF virus that we want to work with. So you can see that it's very multidisciplinary approach. In Pasteur Paris itself, I'm the human geneticist, but we have many virologists, epidemiologists, multiple modelists, animal models, and also we have our Pasteur Dhaka, Pasteur Cambodia, and Pasteur Cameroon as our field site. So in this project, we focus on 3 viruses and plus disease X. Disease X can be anything that can come with our study. So we focus on Rift Valley fever virus, Crimean Congo hemorrhagic fever virus, and dengue. So we have 4 specific aims. The first one is to enhance surveillance in order to detect this unknown virus that can potentially to spill over to human. Second, this is important to understand transmission dynamic of this virus in order to understand how to control them. And third, which is very important, is to understand the adaptation of this virus, because we see that with the COVID, we learned that this is very important for us to fight against them. And the fourth one, which is very important as well, is to understand the adaptive immune response. So this is really the basic knowledge for development of the vaccine. So just a brief explain you about all these viruses that you may not have heard before, or is not very popular. So first is the Rift Valley fever virus. So this is a viral zoonosis. So human got contact by blood or organs, contact of blood organ of infected animals, or even bite of infected mosquitoes. There's no human-human transmission. So you can have a complication, you have loss of sight, confusion, bleeding, liver problem. So the only way to control this is to have sustained program on animal vaccination. So this is the area in Africa that we have our endemic and epidemic of Rift Valley fever. So the second one is the Crimean Congo hemorrhagic fever virus, CCHFV. So it first described in Crimea, 1944, and then Congo in 1969. So that's why they get the name from that. So it is transmitted by ticks and then from livestock animals, and further is up to 40%. So now we have human-human transmission can occur, but it's mostly from tick. We have no vaccine available at the moment. The distribution, you can see that it's very rare, in Turkey, in Central Asia, also in some part of Europe, and it's so now I heard that it's go to Spain as well, and also in Africa. So our consortium going to use the One Health approach. In these 3 sites in Senegal, Cameroon, and Cambodia, we collect samples from hospitals, patients who get fever with unknown diagnosis. We also collect samples from people living in community close to livestock that we can collect samples, and also we collect insect, tick, to do the study. Our approach is from the field to bench, not bench to field. So we're going to sequence out the virus, understand how the virus adapt, and try to make an infectious clone, and then working on adaptation in animal model. We take samples. We take isolate B cells specific for that virus, and then we isolate monoclonal antibodies and try to understand the function in terms of neutralization of this antibody to the virus. We're going to select T cell, doing T cell mapping, and to understand the function, we're doing RNA sequencing, single cell RNA sequencing to understand the immune response and pathogenesis. And we create a big biobank of sample that we collect from animals, insect, and human with different phenotype. So I just show you some prelim data, which is I think very interesting. So this is the work that we done in Senegal. We have we have 24 sentinel sites across the country that we collecting patients with fever. And here we screen for arbovirus that circulate in that region. You can see that it's interesting. The blue one is dengue. So I showed you that before that dengue is not in Africa, but now dengue is the most common arbovirus in Africa, not yellow fever anymore. So it's like dengue is spreading around the world. What also very interesting is this one, when we, with basic aim to understand transmission dynamic, because we collect samples from cattle, sheep, and goats, animals, livestock, and human samples. And we're looking for antibody against Rift Valley fever and CCHFV. And you can see that the blue one is the CCHFV serology positive, is nearly 30% in some area. So it means that this virus already circulated in the livestock. And in human, we can see some prevalence, which is that is already spilled over to human. So even you don't have the epidemic, you still have silent transmission of that virus in the population. So if you really want to prevent the pandemic, you have to go very early and then try to stop break the cycle of transmission in this animal and human population. Even they get sick because we can see that when people get sick, it's always too late to control. So I come to end because as you know, I also come from Japan. I worked in Japan for 3 years. So my objective mission in Japan is to create Institute Pasteur Japan, which I come here in order to see whether we can set up some collaboration with Institute Pasteur Korea or other Korean researcher. So this is the project that we plan to have in next year. So just to briefly introduce you, the construction of Institute Pasteur Japan will not be like one big building and research center competing with other research institutes in Japan. It's not going to be very difficult for us. So we're going to create a multidisciplinary group of researchers, Japanese, French, and Pasteur scientists to solve the world important problem. So the first center that we're going to do is the called Planetary Health Innovation Center, which means it's a global health, one health, but then we also including environment too. We're going to do, try to restore biodiversity, but SASH also show that biodiversity is one of the factors that causing emerging infectious disease. Also the clean energy, clean air, nature-based solution for agriculture, food preservation, food solutions, green city, rewilding, or vector-borne infection will be this one. So the second one will be focused on aging. This is a very important problem in Japan and will be a problem around the world. Also, the collaboration with the National Center of Global Health and Medicine and National Institute of Infectious Disease of Japan will be focused on this pandemic preparedness. So the other one that very important that you can see that about social life science, because we cannot eradicate or we cannot do anything without the involvement of politicians, local health authorities, and also the public communities. So this will be a research center that will bring in social science and life science together in order to work together. So the plan that we can see from Dr. Jerome Kim, we have lots of vaccine, but we can eradicate only one virus, which is smallpox. And why we have the efficient vaccine and we cannot eliminate or eradicate the other virus? This will be the subject of this center, because it involves all this social life science. So we already signed this agreement with the University of Tokyo for the creation of this Planetary Health Innovation Center. Actually, the scientists that are going to work with us come from Faculty of Agriculture and Life Science of the University of Tokyo. So this is something that I would like to thank you about. This is also the plan that we're going to work together and how we could work with you and also with Thailand and with all of the network that we are here. So thank you very much for your.
Thank you. Thank you, Professor Anavaj Sakuntabhai. It's really great news to pleasantly listen to your work as well as the establishment of Institute Pasteur Japan. I think that it's going to be a very strong neighbor relationship. So is there any questions on the floor? I think that we don't enough time, but I think yes, please.
Thank you very much for your. Actually, we wanted to have more about what you are doing. I have one question about for Pasteur Korea, Pasteur Japan. How you make also the connection with the livestock sectors, because with peak rate, actually you show the importance of the livestock in terms of the disease, Rift Valley, Crimea Congo. So is it also a connection with the livestock department?
Yes, that's a good question, because when I arrived in Japan 3 years ago, I think that my mission is to work on this One Health. And we found very difficult, as you see, that One Health is not only one ministry. And the way that people work also, because if you want to work with livestock, you have to go to Ministry of Fishery and Agriculture of Japan. So the way that I'm trying to do, because when we talk to them, they don't care about human health, right? Also, Ministry of Health, we would not care about animal health. So what we're going to do is we're going to go through scientists, and then scientists will be working up to the ministry. So now I already discussed with you now that on this Saturday we're going to have a meeting to discuss about eradication of Japanese encephalitis virus, which is prevalent in pig population in Japan with 80% positive. I think Dimitri will have some idea about in Korea how many pig. You also have many pig farm, right? So then my researcher contact a pig farm in his region, and they agree that we can do sampling, from that pig farm, and also we're going to collect mosquitoes in the farm. And also because our Japanese ancestors can be in wildlife as well. So we can contact the Association of Hunters because when they do hunting, they can give us samples. So this is how we can collect samples from livestock and also we can do our sample from hospitals or people in the life in the farm. So this is our approach. We're doing all these operations. So I'm going to collaborate with you because we are only biologists, and we would like to understand the effect of environment, ecosystem.
So we need to move to the next speaker. Thank you, Professor Sakuntabhai.